So we're going to talk about cells now, and we're also going to talk about their parts, the organelles. If you look at that word, organelle, it has the word organ in it. For you, you have heart, you have a brain, you have kidneys, lungs, all sorts of organs. They are structural units of the body that perform a certain task. Like your heart, for example, it beats, it sends blood through your body. Your brain is responsible for controlling your body. Your lungs pull in oxygen, and so each organ has a job. What we're going to learn about in cells is that they also have organ-like structures, but they are very tiny, and so we add this to the end. E-L-L-E-S. That means tiny. And so what organelles mean is tiny organs. So we'll talk about that. So to begin, we're going to talk about the first time that we noticed cells. So the guy who first noticed it is this guy right here, Anton von Leeuwenhoek. And so Anton von Leeuwenhoek developed an early microscope. Now, there were microscopes around before his time. I believe that it was invented by the same people who made glasses. But they developed a microscope, and everybody wanted one. It was like the iPhone of its day, and scientists lined up from, the world around to buy it. And so he couldn't buy one. So he just made one. And this is sort of what it looked like here. It has a little eye hole right here, and you can actually adjust it by twisting it with this little lever. And you would hold it up to your eye, and you would see through that little hole. And so what he looked at was plaque in his teeth, and he discovered what looked like little tiny living organisms that we now know of as bacteria. And so then Robert Hooke was the next guy. Fun fact, Sir Isaac Newton, the man who discovered gravity, hated this guy. Here he is right here. He hated him so much that he burned all of his photos. And so we don't actually know what he looks like. He's sort of lost to history in terms of what he looks like. But he did observe plant cells. And so he took a bottle and he took the cap off. So that bottle cap is made from plants. It's called cork. It is the dried up remain of plants. And so he looked at that under a microscope, and this is what he saw. And if you look very carefully, there's a bunch of like little squares in here. In fact, maybe we can look more closely. Bunch of little tiny squares. Whereas this up here, this would probably be animal cells, I guess, and these are plant cells. You can see that these are round and these are square, and that's because these are plant cells and these are animal cells. Plant cells are square because they have that cell wall. They have an extra layer. Okay, so he looked at those cells, and he called them cells because they look like the architectural drawings for a church monastery room, which was called a cell. And so that's where we got the name cell from. After discovering the cell, scientists became curious if it was present in all living things. Sure enough, it is. Today we know that having cells is one of the requirements for being alive. But back then they didn't know that, and so they just kept looking at organisms under the microscope, and one by one they showed that they all were made of cells. So Schleiden showed that all plants were made of cells, Schwann that all animals are made of cells, and then Virchow discovered that all cells come from existing cells. One cell can become two. And so this all together helped to create the cell theory, which, by the way, is easy to remember. So just remember that Schleiden, it sounds like sliden. So I like to remember that he's sliding on a banana peel, which, by the way, is a plant. Bananas are plants. And then Schwan is really easy because it sounds like swan, which is an animal. And then Virchow just sounds like Velcro to me. And you can kind of imagine that Virchow here, he took their two ideas and he combined them together. And so the cell theory was created. And the cell theory has three parts. The cell theory was made possible by these scientists. And the first part is that all living things are made of cells. The second part is cells are the basic unit of structure and function of living things. And the third is all cells come from existing cells. And so let's look at these one at a time. All living things are made of cells. This means that no matter what you are, if you are alive, you have cells. The next one is they are the basic unit. This means that anything smaller than a cell is not alive. And so if we look at this organism here, this is a colony of algae. And so each one of these is a cell. If we look at these tiny little green dots, that's actually a type of organelle called chlorophyll. And that is not alive. Yet the cell is alive. This is actually an interesting organism. It's called Volvox. It is a collection of algae. Each one of these cells used to be an algae cell, and they come together. And we've actually seen that this can become a multicellular organism. And this kind of proves that single-celled organisms evolved into multicellular organisms. Of course, you are a much more complicated version of a multicellular organism. Volvox is just a very simple example. Okay, and then all cells come from pre-existing cells. That kind of just goes with biogenesis. Living things make living things, and they do it through cells. So, for example, you were first made of one cell that was called a zygote. That was the first cell that you ever had. And that zygote divided, and it became a blastula. And that kept dividing, and it eventually became you, with all of your trillions and trillions of cells. And so then that begs the question, if all cells come from existing cells, where did the zygote come from? Well, it came from two cells. It came from a sperm from your father and an egg from your mother. And so it did come from a pre-existing cell. Cells are limited in their size. We don't find really, really big cells. And the reason why is because as cell size increases, the ratio between surface area and volume decreases. And so, for example, if instead of cells we consider these cubes here, if you look at this cube, it has a certain surface area. A surface area is just all of the spaces that can be touched. And so, for example, I can touch this surface, I can touch this surface under here, over here, and in the back. Those are all surfaces that can be touched. The volume is going to be the same for all of these. The volume in this case is the amount of blue material. And so by cutting this material in half, we are not losing any of the volume. It's still the same, because if you put it back together, it would still be the same amount of material. The difference is we have now created this surface and this surface underneath. And so what we have done by cutting this object in half is increased the surface area. And so the more that we do this, the more the surfaces increase. And that is why cells are so small, because it increases the amount of surface area. The more surface area, the better per volume. And so if we look at the ratios here, we see that for every 1 centimeter cubed of volume, there is 3 centimeters squared of surface area. The greater the ratio, the better. So over here, this ratio is much, much better in terms of surface area. Why does a cell need surface area? Cells need to have enough surface area to allow sufficient materials to move in and out of the cells. And so, again, surface area is just all the surfaces that can be touched. The more surfaces, the more something can absorb a thing. And so when I say a thing, I mean, of course, materials. And so what are materials? What materials does a cell need? A cell needs things like oxygen to flow through it. It also needs things like water and food. You need to feed your cells. And so the more surfaces that you have, the more that absorption can occur. If your cell was really, really big, then there would be certain parts of the cell that would never be reached, like this part that I'll draw in green. That would never be reached. And so the cell would not be filled with those nutrients. We need smaller cells because they're easier to feed. All right, so we're going to kind of wrap this up now. This is the last part of this lecture. And as I leave you, I just want you to remember that when we talk about the parts of a cell in the next lecture, that the structure of them determines how they function. It's not magic. There isn't just a need for something and the cell magically makes it happen. Everything inside of a cell is structural in its nature. In the same way that this bicycle tire has to be the right shape for it to perform the right function, so too does a cell and its components. And when they are not the right shape, they don't perform correctly, like this square bicycle tire. Now he can get by, as you can see, but it's really, really hard. And the same thing is true for us. When we have cells that aren't quite the right shape, or even smaller stuff than that, like proteins, when proteins are not the right shape, we have problems. And so let's just go to that. So proteins have a very particular shape. Like here's an example of a protein. And maybe the job of this particular protein is to grab something and pull it into the cell. Maybe that's its job. And maybe what it's pulling is, maybe it's pulling in salt so that water can follow that salt and we can have proper hydration, right? So this protein normally would hydrate you, but if you lose that structure through this process here, it's called denaturation, you're going to lose the shape. And as you can see, this protein has lost its shape. Now normally, the flow of information is you have your amino acids, and amino acids are joined together to make polypeptides. And then those polypeptides fold to become proteins. However, if you unfold them, they become polypeptides again. And so that's what we see here. Through denaturation, this protein here has unfolded and become denatured. And so this is really just a polypeptide again. And it won't work. It will not do the job. It's a lot like that square bicycle tire. And so, for example, in this person, if they're not making the correct protein, they could become dehydrated and die. And so that actually could really happen. So what causes denaturation? Well, if you overheat your body, if you get too hot, if you were to jump into a vat of acid, if the pH is wrong, ions can do it. So, for example, if you are electrocuted, radiation can do it if you are next to a nuclear bomb as it explodes. These are all examples of ways to denature proteins and make them lose their shape. Now, you can renature sometimes. You can renature by going back to homeostasis. And so the bottom line here is a normal protein must be in homeostasis. And denaturation occurs when you're out of homeostasis. This is a big part of the reason why we have to, for example, have the correct body temperature, because stuff like this would happen. And you need your proteins to perform their job. All right, so that's going to be it for this part of the lecture. And next, we're going to talk all about organelles. So I'll see you in the next lecture.